trials, the recent publications that led to what is our current standard therapy, and then we'll sort of rally at the end with a little bit of discussion about relapsed disease and considerations related to that. So, from this slide right here, what I'm trying to illustrate is that, not all lines are the same. These are all lines. Hofmeister's probably going to say one of those is a segment, but I'm going to tell you they're all lines, okay? And you can see, they have different implications, right? When you see the police line, right, you might be worried. When you see the line right above that, you're not quite sure where it ends, right? And that actually, it's relevant in myeloma. We do have a definition for what a line of therapy is, but the definition doesn't really tell the whole story. So when we say there's a predefined course of therapy that uses some combination or sequence of drugs, either together or in some planned order, without needing to stop the therapy because of toxicity, and without needing to switch the therapy, not because you were planning to switch it at a certain point, but because the disease is progressing, that's all one line of therapy. And so these are examples. So we talked about a frail standard risk patient, and we talked about lenalidomide, dexamethasone, Rev-Dex, being reasonable frontline therapy. And we generally use that for as long as it's working, right? You could also, in a younger patient, use that same induction therapy for 3 or 4 or 6 cycles, move into transplant. So that patient gets high-dose melphalan as part of their therapy. And in this example, maybe you stop after that. Let's just say we talked about the low-risk patient in complete remission. So let's say that patient doesn't get maintenance therapy, right? And then we have, I think, a very standard frontline sequence of Rev-Vel-Dex, RVD, transplant, and then lenalidomide maintenance. Some people may get lenalidomide dexamethasone maintenance. I don't typically use dexamethasone. There are some exceptions, and we'll talk about that. And then there's the next line of therapy illustrated here is total therapy. Anyone visit Arkansas? Anyone consult at Arkansas, right? So, the total therapy program has been this sort of evolving series of regimens where, as new effective therapies come into being, they get incorporated into some prolonged, planned course of therapy that currently is about 3 to 4 years long and involves triplet maintenance therapy. So that's total therapy. And as you can see, these are all really, really different. These patients haven't just gotten different lengths of therapy, different intensity of therapy. They've also been exposed to very different drugs over the course of their therapy. And so it's fine to say, well, we have 4 patients who've gotten frontline therapy, but when they need second-line therapy, they're not necessarily at the same place. Does that make sense? So it's a shortcoming in our definition of what a line of therapy is. But what I want to do is I want to focus on Rev-Vel-Dex, transplant, and Len Maintenance. And I want to actually walk us through how each part of that line of therapy actually became the standard that we use today, right? So let's start with the beginning. Why RVD? Well, there was a randomized study that compared RVD to just Len-Dex, and it was shown to be superior. This was a 500 and something patient study, and the primary endpoint was how long does the disease stand under control before it progresses? That's progression-free survival, and the difference was about a year between these. And it's interesting because the addition of bortezomib in this trial in the top box really was only during the induction period. The maintenance therapy for these patients was the same. Everybody got the same lenalidomide dexamethasone maintenance on this study after some period of induction. And so just adding that third drug for a finite period of time ended up leading to more prolonged disease control. The overall response rate was also higher, about 10% higher, 82% in the 3-drug arm. A little bit lower than previously published. The small original phase 2 studies showed actually, the first one of RVD showed 100% response rate, but I think any doc in a myeloma clinic knows that the response rate to RVD is not 100%. It's probably a little bit higher than this, but it's 90-ish, 90%. And most importantly, the chance of getting into a complete remission was just about doubled when you added the third drug. There was a small difference in survival, but patients lived a long time in both arms. And survival is a very difficult thing to measure in frontline studies. And the reason is, as I'm going to show you later, we have really good second-line therapy and really good third-line therapy. And so when you do, it ends up being a bit of an equalizer. And so what drives survival usually often isn't exactly how long you respond to frontline therapy. It's how long do you respond to your first 3 lines of therapy or your first 4 lines of therapy when you add them all up. Actually, there's been quite a bit of work looking at what we call PFS2, which is progression-free survival for 2 consecutive regimens added together, things like that. Just a bit more complex way of looking at efficacy of different sequences. So this was the winning arm. I mean, really, in every parameter that we looked at, RVD was better. There was more toxicity, though. I put the bortezomib at the top in italics because in this particular trial it was given in a way that we don't give it. That's IV therapy twice a week. That's how it was originally given, and that's how it was originally used in the first publications, but we don't do that. And the reason we don't do that is because when you give it this way, the incidence of severe neuropathy compared to the other arm was 3 times as high. Okay? And so to minimize the toxicity and balance efficacy versus toxicity, we've modified these regimens, and the Craig mentioned that Dr. Hoffmeister mentioned that there was an RVD and there's an RVD light. And basically what the difference between those is the schedule and route of administration, more or less, of the bortezomib and steroids to some effect. So that's how we got to the induction part. Now we consolidate with transplant, and why do we do that? Because, well, actually because of numerous studies over years comparing transplant to no transplant. But the latest and perhaps most important is a French study that was just published this last year. And the reason it's important is because it's one of the first large randomized trials that addresses the question of timing of transplant after what we consider to be modern 2017 therapy, right? And we heard about the American study that actually had an identical design to this except for the duration of maintenance. We were all participating sites in that study, and this is the French counterpart to the study, and they were originally designed actually to be run together on both sides of the Atlantic and pooled. But that's very hard to do, actually. So the French actually completed their study and have published preliminary results already. And their results reflect this design: Rev-Vel-Dex for 3 cycles for everybody, and then stem cell collection for everybody. And then half the patients go on to transplant and get a couple more cycles of RVD after that, and then maintenance. And the other patients freeze their cells, don't get the transplant right away, and go on to just get more RVD. Everybody comes out the other end and starts maintenance therapy, 15 milligrams of lenalidomide a day for a year. Now in the American study, it's indefinite therapy until the patient progresses. And so we will still be able to use these studies in a complementary fashion to start looking at the question about optimal duration of maintenance therapy. In this particular study, patients got treated for a year. And so in the MSMART criteria that we saw before, they mentioned a year of lenalidomide maintenance. Well, this is the evidence that a year of lenalidomide maintenance is based on, because the study that's been completed, or the half of these paired studies that have been completed, used a year of lenalidomide maintenance. And what this study basically showed is that early on out of the gate, there's benefit to transplant in terms of disease control. Progression-free survival, again, over a year longer when transplant is included early on. Also, complete response rate, about 10% higher when transplant's used early on. But survival is the same for the reasons that I told you before. Survival is not different in either arm. And actually, just to put any of the other survival data in perspective, the survival at 4 years was 81% in both arms on this study. Okay? So transplant or not, when you have patients under the age of 65 who are fit enough to be transplanted, who get either one of these therapy routes, the expectations are quite different. When I started, the 1 year survival was 75 to 80%. And now the 4 year survival is over 80%. So much progress. The study also looked at something called minimal residual disease negativity. And what that means is using extremely sensitive techniques for finding amounts of myeloma cells well below what we can see with a microscope in a bone marrow biopsy. Minimal residual disease negativity also occurred more commonly in the transplant arm, but it did occur in both. And when it occurred, it's a really good thing. The deeper the suppression, it's kind of intuitive, but we haven't really been getting patients into that deep stage until recently, right? So now what we see is the deeper we suppress somebody, the better it is. And transplant's more likely to achieve that. So I think this study, more than any, really solidifies the recommendation for early transplant. But because of that lack of survival benefit, it does leave the door open for a deferral of transplant until later for patients who opt to do that. And finally, the part about maintenance. Why lenalidomide maintenance? Well, there's been several studies. These are actually only 2 of several studies that have looked at lenalidomide maintenance. And I'm not even going to talk, honestly, just in the interest of time, I'm not going to talk about bortezomib maintenance, although we heard that bortezomib maintenance probably is actually important in certain chromosomal high-risk subgroups. But in terms of len maintenance, the more common drug, we have the CLGB study, a randomized study, which after whatever induction regimen and transplant the patient gets, patients who've had at least a partial response then get randomized to placebo or lenalidomide. And this was a 450 patient study, and basically what we saw was a doubling of the progression-free survival when you do maintenance compared to no maintenance at all. A doubling. Okay? At this point, it's somewhat difficult to do non-maintenance, placebo-controlled maintenance trials because you have data that's this strong. The only subgroup you can really do that in easily are patients with sort of low-risk disease who have very good disease control going into the trial. The other study that I showed here is a European study, and in this there was actually a randomization up front, so there were a couple of different induction regimens, but then they all went to transplant. But they were randomized against transplant, and then after that, whether they got transplant or no, there was another randomization to just lenalidomide or lenalidomide plus steroids, and the steroids were 50 milligrams of prednisone every other day. And we know that when you're using IMiDs, when you're using Revlimid, when you're using Pomalyst as active therapy for progressing myeloma, we know that the response rates are higher when you add steroids to the mix. In the case of Pomalyst, it's like 3 times higher for relapsed disease when you add dexamethasone. So I think it is a very important question to ask whether a relatively tolerable drug, I mean, we know about steroids, but whether the addition of steroids makes maintenance therapy better. And actually what this study showed was no. There was a slight improvement with the prednisone, but it wasn't statistically significant, so mathematically you couldn't rule out that it was chance. And so we and others generally don't use dexamethasone as part of maintenance therapy because of that type of data. So the question that everybody wants to ask right now, I mean, I gave you data that says transplant helps with early disease control, but I also told you at the same time it doesn't improve survival, at least in that study. And so the question is, can we just get rid of it? I mean, I get asked that question all the time. Can't we just get rid of it? And so the exercise, anybody who's been in my clinic and asked me that question has gone through the following exercise. I tell them, imagine writing down all of the therapies that are approved for myeloma on a card and spread them out on a table, right? And then after they do that, I said, did you really do all of them? Is there anything else you'd want? And they say, well, I've heard about immunotherapy. And then I said, okay, are you on curcumin? And they say, yeah, I'm on curcumin. And I probably would want access to that stuff too. And so they spread them all out on the table, right? They're all out there. Okay? That's a joke, but I'm actually pretty open-minded about alternative and supplemental medicine. I'm teasing. But the point is if you took this and then you told the patient, okay, now pick one or two of these, take them off the table, and throw them in the garbage, right? I mean, if it was Velcade or Pomalyst, you'd be ticked off, right? I mean, you wouldn't even accept that. It's like, why on earth wouldn't you make Velcade available to me? Think about transplant. What does the French study that I showed you tell us? It tells us that transplant is a line of therapy. It can be part of the first line of therapy, or it can be a later therapy after progression. But if you just skip it, if you throw it away, you're basically depriving yourself of a line of therapy, right? And it's the one line of therapy out of all these that you really have to have some vigor for. So not everybody's a transplant candidate, but if you are, waiting until you're not to discuss it again is crazy, right? It's something that you probably will want to think about using while you're healthy enough to use it, just like you'd use everything else. With salvage therapies, it's easy. When you go back to this picture, you can imagine just, sort of like peeling away salvage options, and it's really easy to see how survival might get lower or shorter. You could, this is published. This strategy right here, this is literature, right? So if you do an induction where you peel away some of the, the newer, the Velcade or the lenalidomide, do the transplant, don't do maintenance, wait until you relapse and give like one line of salvage therapy and call it a day. And we call this regimen 1995. That's how I treated patients when I graduated fellowship, okay? And so there's plenty of data about this. And what this basically is current therapy without the best drugs, with less lines of salvage therapy available and no maintenance, right? And survival, as we heard, is 1/2 to 1/3 as long as it was in 1995. So should we get... What? How many transplants can you have? Well, it depends if you're being treated at Arkansas or not. Most patients never have more than 2. Okay? And I can't speak for Michigan or Ohio State, but I can tell you that although we collect enough stem cells for 2 transplants on every patient we can, the majority of those second bags never get used. For a variety of reasons, but they never get used. So it's an insurance policy of sorts that's never cashed in. Okay. Another question that comes up is, what about an allo-transplant? I've heard allo-transplant's a more potent type of transplant. It's a donor transplant instead of your own cells using somebody else's. Potentially curative because of something called graft versus myeloma. The immune cells in that donor graft are fighting the myeloma. There's some danger, some risk up front because those same immune cells can react against your body tissues. That's called graft versus host disease. As a result, there is a lot of upfront risk, survival risk related to it. And the numbers aren't important. It's the pattern that's important. You're hoping, see the curve flattens out on the allo line there, meaning that some patients are long-term survivors without relapse, they're cured. Fine. You can argue about how high that bar is. At some point down the road, those lines cross, and statistically, you're better off having had the allo. The problem is, everybody in between these curves up to that point, who would have been on the orange curve, but is not on the orange curve anymore, died as a result of the transplant. Transplant-related mortality. And as therapy controls disease longer and longer and longer, that space is getting bigger and bigger and bigger, because the upfront risk with allo-transplant still all happens in that first year, right? And so it's actually getting less and less common. We really reserve it for those ultra-high-risk patients that we heard about, whose expected survival in the first year or two is so tenuous, so risky, that there actually isn't that much of a gap between the risk related to the transplant and not having the transplant. And this is reflected in the numbers. I mean, so in 2015, the Department of Health and Human Services released this data. We did 7,300 auto transplants for myeloma and 190 allo transplants in the whole United States. Right? So that's you know, a third of the myeloma patients diagnosed that year got auto transplants, and hardly anybody got an allo. Right? So this is pretty universally, I think, agreed upon. And now just really just a couple minutes, because there's this relapse disease and salvage therapy really becomes the Wild West. You know, I mean, it's because there's such heterogeneity in what we do first line and then second line. By the time you're at third line therapy or beyond, you know, you're really starting to come up with patient-specific things. So these are actually approved combinations, FDA-approved combinations of drugs, and the ones in italics are the ones that were the winners of randomized studies that led to their approval. Pomdaro was also, Pomalyst and daratumumab recently approved on the basis of just phase 2 data, I believe. So how do you choose? I mean, like, yeah, there are so many options here, like, how on earth does a patient end up on one of these? And it ends up being, you know, patient-specific, and to some extent often patient-driven. What have you had previously? How well did it work? Did you actually progress while you were on a specific therapy or long after you took a break from it? And how long ago was that? What kind of side effects did you have from that drug, and do you still have any of those side effects? Neuropathy is one of the ones I'm thinking about. What other health problems are in play? You know, do you need a steroid-light regimen because your blood sugar is completely out of control? Do you have to use an extremely low dose of bortezomib because your neuropathy is out of control? These are considerations. Another one is always, comes back to this other question about the number of transplants. Are there stored stem cells? And again, just as the first time around, you still have to have a certain level of fitness for a second transplant. And then I always ask the question, is a patient a candidate for a clinical study? This is an actual exchange I had with somebody on Twitter, and one of the problems of being completely anonymous on Twitter is that Myeloma Sucks might be in the room right now, and if you are, I'm so sorry. But I'm actually online with my picture on Twitter, and so somebody just threw the question out there, you know, what's the biggest obstacle to cure? And I said, we don't put enough patients on trials, which leads to slowing of drug development. And then I got an earful about being a guinea pig, right? And that is an extremely common sentiment. It just doesn't usually happen to me online. But you have that conversation with patients, and there's, you know, patient attitudes, patient concerns about being the subject of a experiment, heightened by the Henrietta Lacks movie that came out in the last year, and the book that came out a few years before that. I mean, really is real. I mean, I think that it's I think hard for patients to understand how many checks and balances there are in human research because of all the abuses that have happened in the past, and we're trained about research, and we're very conscious of those issues. There are also other things that might, you know, affect the likelihood of a patient getting on study. All of these kinds of things like severe neuropathy or bad kidney function or low blood counts, those are the kind of biological things where the exclusion and inclusion criteria for a study might just say, you know what, you don't qualify for the study. You want to be on it, I want you on it, you don't qualify. There's also logistical ones. I saw a patient yesterday who's the perfect study candidate. the study that we want to put him on, there's just not a slot right now. And we're waiting. Hopefully, in the second week of October, there'll be a slot. Or there won't be. I mean, so it's that we're, centers scrap with each other for slots, patients scrap with each other for slots. With targeted therapies, increasingly, screening for the target is going to reveal that you don't have the target for the drug that you're hoping to get, and then that doesn't make any sense. And then also things just really easy things like having to travel or not having insurance coverage for clinical trials sometimes actually ends up being the thing. So when patients can't end up on therapy, then instead of adventures on Twitter, it's adventures in therapy. And this is just I did this slide last night, and I was just thinking, what are all the things that you'd never read about in a textbook that I've actually tried on patients? And most of these, some of them are actually what one of my attendings when I was a trainee called French chefing, where you're a pinch of this and a pinch of that and a dash of this and stir. Most of these, though, are actually based on publications, specific publications, but they're like phase 1 and phase 2 studies where you have 30 patients, 50 patients, 12 patients treated with a given regimen. You know that you know what's been used before by others, you know what to expect in terms of side effects, and go. There are some of them that I use more commonly than others. Carfilzomib, pomalidomide, and dexamethasone, super active regimen. Pomalidomide and elotuzumab is an active regimen. Pomalidomide and DARA was on the list, and I took it off because it was recently approved, so it doesn't count as making things up anymore. And then Benda-Rev-Dex. Bendamustine is a drug approved for chronic lymphocytic leukemia and lymphoma, and it's active in myeloma. It's in the same family of drugs as melphalan and Cytoxan, and it is combinable with Revlimid and Revlimid or Velcade, and we actually published a paper combining it with Revlimid. Very high response rate, very hard on your blood counts though, so use with caution. So to conclude, the frontline paradigm of induction, consolidation, maintenance, that's here. That's been here since total therapy. It is now universally adopted. That's not going to go away anytime soon. Whether consolidation has to include transplant may not remain the case, but for now, based on the latest available data, I think transplantable patients should think very hard about transplant early. We are gradually refining each part. Triplet regimens, as I showed you, we use RVD frontline, but we're looking at other triplets frontline, and almost all of those options that I showed you, the approved ones and the phase 1-2 type ones, those are almost all triplets. Okay, and so we still use triplets, and they tend to work better than doublets, even as second-line therapy. Response rates remain in the 80 to 90% range for second-line therapy and sometimes for third-line therapy. So we expect disease control. And finally, I'm a big proponent, as you know, of clinical trials. And it's particularly because of that sort of spiraling cycle of flipping from drug to drug to drug. When disease is beyond third or fourth line of therapy, we really should think hard about trials. And as my Albanian wife taught me to say, falemderit, thank you very much. And I hope if you have any questions, I'm happy to answer.